Hi there, I'm Brooke Wolford. I am an incoming postdoctoral fellow at the KG Epson Center for Genetic Epidemiology, and I'm excited to talk to you today about the functional characterization of GWAS results, which is just another way of saying what is the underlying biology at this GWAS locus, which is a common question that we ask ourselves. So first, I'm going to motivate what we're talking about, why we care about this. I'm going to give some intro to genome architecture, and then we're going to dig into a specific locus and kind of think about what the underlying biology is. I'm going to review some data sets that are available and review a few published examples. So first, I wanted to remind you that some GWAS results are really easily interpretable. This is a Manhattan plot, and on the x-axis are each of the chromosomes, and over on chromosome 19, you can see there's one peak for ApoE, and this is a GWAS for Alzheimer's, and ApoE is a known gene associated with Alzheimer's, and in fact, the lead tag SNP is a missense mutation that changes the protein. And so this is very clear cut what functional impact is therefore affecting the outcome, which is Alzheimer's. But then there's a lot of loci that we find that are a lot more complicated. So this is an example from the FUSION study, which is studying type 2 diabetes. And it's about 9,200 people from MetSim, which is a Finnish data set. And this is a locus zoom plot. So the x-axis is just zoomed in on the genomic coordinates, and the y-axis is the p-value from the conditional analysis from GWAS. And so this is a GWAS for a quantitative trait at adiponectin levels. And adiponectin is a protein encoded by the gene ADIPOQ. And so you can see here at the bottom that ADIPOQ is right here, and there's all these signals localized above it. But there are 7 conditionally distinct signals found from this stepwise conditional analysis, with A being the most significant, and then moving down through the alphabet. And so what's going on here, if these are independent, how are all of these things affecting this gene or nearby regions? That's the question, and that's what we're going to focus on today. So let's dig into some things about genome architecture. You may be aware that we think of the genome kind of linearly stretched out like this, and we think of gene regions, genic regions, and then intergenic regions, which are also non-coding regions. And so I've used these colored blocks to indicate different parts of the gene. So we have our enhancer, which is typically nearby a gene. Then we have our promoter, where our polymerase is going to bind. We have the 5' untranslated region. We have some combination of exons and introns, and the introns are ultimately spliced out when we transcribe to RNA. And then we have the 3' UTR, and then I'm kind of continuing on to some next gene down the line. We think of these exons as the coding regions, and anything else is the non-coding regions. So for us studying GWAS, we spend a lot of time thinking about the non-coding regions because the majority of GWAS loci are in fact in these non-coding areas. And so it's not as clear cut how they affect the gene. When something's in a coding region, you can tell whether or not it's going to change the RNA and change the protein and change the protein's function. But when we're looking in these non-coding regions, it's a little less clear. So here I'm just showing the fraction of GWAS tag SNPs from the GWAS catalog for all traits and for type 2 diabetes on the right. And you can just see the vast majority are in non-coding regions. And I've sort of already defined these parts of the DNA for you. The things that we really think about when we think about regulatory regions are the enhancer and the promoter. So places where the transcription of the gene is being altered. And that can kind of happen in a more 3D space if you think of the DNA looping and having this 3D structure or 3D conformation. And it actually brings transcription factors, which bind to transcription factor binding sites in enhancers. It brings them closer in proximity to some transcription start site where the RNA polymerase is going to bind. And so that enhancer that I've drawn here could actually be megabases away from the actual gene that it is affecting, because there's all this looping of the DNA. So it doesn't really have to be super close to affect the transcription process. And as I mentioned, we think of DNA in this linear way. So the expectation is kind of like a very nice, neat pair of headphones. But in reality, they're actually a tangled mess of headphones. And that's the DNA in our nucleus. And one way that we think about assaying what parts of the DNA touch each other physically is chromosome conformation capture methods. And there's multiple kinds, but basically they help us quantify and identify exactly the areas where the chromatin is spatially connected. So the spatial organization between genomic loci. And here I'm showing a heat map. It's just a little cartoon that's showing these places that are more kind of knotted together, have a higher touching rate. If we quantify the number of times we see them, there's a much more accurate word than touching rate, but it's the number of times that we're seeing these regions interact. And in the places where it's more open, it's kind of a break in this pattern of the squares. So that's going to be useful for figuring out regions of the genome that are interacting. And as I said, there's multiple methods. Ultimately, the goal is to identify the physical interactions between distal regulatory elements and promoters. 3C, 4C, 5C, high C, they're all kinds of chromatin conformation capture. And they really help us figure out what GWAS SNPs in non-coding regions can be connected and correlated to target genes via distance. Even though they're distant, they're interacting. And these are just a kind of a nice summary of some of the different methods that exist. Down here on the bottom, I'll just draw your attention to the real difference is how many of the interactions we're seeing. So 3C is a 1 versus 1, just looking at one area of the genome. And then 4C is looking at one area of the genome, but then seeing what all it touches. And then 5C is many versus many. So it's targeted and it's not just the one region, but it's not genome wide. But high C is the whole genome correlated with the whole genome. So it's very high resolution. And just a quick kind of simplified version of how this works. The chromatin is glued together basically, and then pulled down by an antibody, but it has now linked together these parts that are distal but touching within the nucleus, and then it can ligate those two pieces together and ultimately sequence it or use a microarray depending on the technology to read the two pieces that are linearly separate but are now put together because of this technology. And just to remind you how our DNA is packaged up into chromatin at this big level that we're used to seeing, it is a kind of X-shaped chromosome, but really the DNA is very wrapped up in there into a chromatin fiber. And that is a bunch of DNA wrapped around the histones. And whenever there are 8 histones, that makes a nucleosome. And you might have heard the phrase beads on a string. That's sort of how we would describe what this looks like. But when you really unwind it all the way, it's just a double helix. So these histones are the proteins that the DNA wraps around. The nucleosomes, we know that the DNA is wound around it 1.65 times. So it's a very standard winding process. And then the chromatin is just the complex of the DNA and the proteins together. And that forms within eukaryotic nuclei. There's a few molecular assays that we can use to, again, quantify and sequence the regions where chromatin is open or closed. So when something is more tightly wound up into this histone nucleosome structure, then we call it closed chromatin. And it's not very accessible by RNA polymerase or by transcription factors. When it is less tightly bound, it's more of that beads on a string kind of look, which I'm showing here in this illustration. And there can be some activity into the DNA that's between these nucleosomes. So here I'm showing a transcription factor that has bound here between these nucleosomes, and DNase is an enzyme that cuts DNA. So you can digest DNA with DNase, and it makes cuts all along any of the open chromatin. And so then you get what's called a DNase hypersensitive site. So it's an area that's very sensitive to it. It can be cut really easily, and we would get a peak in reads. So if you're making cuts here and then sequencing where you've made cuts, we're going to get a lot more peaks in places that are sensitive to the DNase and therefore very open. But we're going to get fewer peaks in places that are protected, either protected by nucleosomes or protected by transcription factors. And so that's what's happening here. There's a bit of a dip in the signal. And this is just an illustration, sort of quantifying what a DNase peak would look like from DNA sequencing. Another similar assay is ATAC-seq. This stands for Assay for Transposase Accessible Chromatin Sequencing. And so rather than using DNase, the enzyme is transposase, TN5. Transposase does a little bit better cutting between the histones, so or the nucleosomes rather. So that is happening here. And again, it's cutting between anything but being more protected by transcription factor. So we see something similar here that we saw for the DNA-seq, but we're also getting some signal in areas that are open between the nucleosomes. And actually, it's been demonstrated in the original paper about this method, Buenrostro et al. From 2013, that you can see this pattern of the nucleosomes, and you can see a single nucleosome and then a dimer, a trimer, a tetramer. So there is this decreasing amount of read density, but it's counting like how many nucleosomes the DNA is wrapped around. And the fragment length corresponds to what we would expect for DNA to be wrapped around the nucleosomes. So outside of chromatin, we also have epigenetic levels of modifications. And the two that are the most interesting for us are DNA methylation. So that's actually a cytosine within the DNA that has a methyl group attached to it. And then the histone code. So I mentioned that nucleosomes have 8 histones, and here they're labeled H2A, H2B, H3, H4, and H1. And there can be chemical modifications put on each of these. And biologists have found patterns about if it's an acetyl group or a methyl group put on which specific histone and what that correlates to functionally in the genome. So there's several methods for quantifying and finding areas of DNA methylation. And methylation is typically associated with silencing gene transcription. It's associated with more closed chromatin. And the DNA methyltransferase is what adds a cytosine residue to DNA, and whole genome bisulfite sequencing is a method that allows us to find the areas where those methyl groups are and quantify how many reads we're finding there through sequencing. And there's a bead chip that performs this, or in addition to the sequencing, there's a bead chip that is a more array-based method that allows us to quantify methylation, and that chemically works different than the whole genome bisulfite sequencing. The method for measuring histone modifications, which are indicative of chromatin activity, is called ChIP-seq, or chromatin immunoprecipitation sequencing. And this is performed using antibodies that bind to specific proteins. So it can be a protein that specifically has a chemical modification on it, like an acetyl group. So in this illustration, I'm showing an antibody binding to H3K27 acetylation. The K here stands for a lysine, so it's a specific numbered lysine on the H3 histone, and it's an acetyl group that's there. And then you get reads by some, numeric quantification. They're higher right at the density of the reads is higher right at the part of the DNA where the acetyl group is bound. But you can do this for other things. So transcription factors, which are very commonly the binding sites are in enhancers, and so you can pull down the DNA that's attached to a transcription factor that's actively bound, or RNA polymerase, which is getting ready to perform transcription and bound to the DNA. So the readout from that can be useful for a variety of different proteins that are bound to DNA. So let's put it all together. I mentioned earlier that there's kind of open and closed chromatin, and that there's these enhancer regions with transcription factors. So we can look across the whole genome and identify regions that correlate with a specific state. So a repressive state or an enhancer state. And we can use all the things we've learned so far. So we can look at chromatin accessibility with either a TAC-seq or DNA-seq. We can look at if a transcription factor is bound with ChIP-seq. We can quantify the transcription factor binding site via other methods, in silico methods, and sequencing that lets us, see what genetic variation exists there. RNA polymerase with ChIP-seq, different histone methylation, acetylation, and ultimately we just boil this all down to a chromatin state. So here we really only have a trimethylation at H3K27 and no chromatin accessibility peaks. And so this is a repressive state. It's very tightly wound DNA that is not very accessible and nothing's really happening there. But we have an enhancer state here where it's an open enhancer or open chromatin. Transcription factor is bound. And then there are these different marks like H3K4 trimethylation, monomethylation, and H3K27 acetylation, which are known to be correlated with enhancer activity. So we call this an enhancer state. We also have promoter states, which vary a little bit with these different histone modifications and then also have RNA polymerase bound to them. We have a transcribed state, which has H3K36 trimethylation and then also the RNA polymerase bound. There's poised state, which means it's kind of almost ready to be transcribed. And in this situation, there's this H3K27 acetyl is lacking. That's associated with active enhancers. So it's like this is an enhancer region that's poised and ready to be transcribed. And then again, we have this repressive state. So we can look at windows along the genome and call them different annotate them as different states based on all of the information that we looked at so far, all the different molecular assays. And this is done via a chromatin hidden Markov model, Chrome-HMM. And you can tell the algorithm if you're looking for a certain number of states, so like 10, 11, 12. This paper that I'm showing a figure from, they identified 15 states, and then they went through and named them based on the different characteristics that they had. So similar to what I just showed you, this bivalent poised transcription start site has this H3K trimethylation on the H3K4 trimethylation. There's a couple different transcription states. They vary a little bit. There's an active transcription start site that has this H3K4 trimethylation. This enhancer region here has they don't have the K27 acetylation here, but we know from other experimental assays which of these characteristics match, and so they can go through and name these different chromatin states. And it's an unsupervised learning approach that allows you to label at the end, but ultimately allows you to define all of these annotations along the genome. And then this paper did this from the Roadmap Epigenomics Consortium, and so they looked at how much of this annotation overlapped things like exons, genes, introns, transcription start sites, how much it overlapped expressed genes or repressed genes, and it all made sense. Like these different states made sense with what we know about these different types of genes. Also with methylation, the CpG percent methylation was less here at these active transcription start sites, and a little stronger. CpG methylation is a specific type of methylation that exists, but it's the strongest here in this repressed region of the genome. So that all makes sense. DNA accessibility is higher in these active transcription start site regions and in enhancers. And then also in looking for enrichment for conservation across species. So are there certain types of chromatin states that are more or less conserved across different mammalian species? So this is just a nice kind of putting all of these different pieces of information together to understand the genome architecture. Kind of zooming in on what I just mentioned for this GERP score. It's a conservation score and it's calculated per base, and it's looking across different species to see how aligned that region is across multiple species. And if the number is greater than 0, it indicates that fewer than expected substitutions have been made, so it's evolutionarily constrained. And then if it's less than 0, it indicates weak evidence of accelerated rates of evolution. So here I think they've actually shifted from 0 to 1 is what we're interested in looking at here. But constrained elements are just regions that are highly conserved, and we are interested in things that are conserved because they're likely important. If it's important in me and in a zebrafish, if it's conserved, it's probably doing a really important like housekeeping function, like a housekeeping gene that does something broadly, like a ubiquitinase that ubiquinates a lot of different proteins in our bodies, or something more biologically related, like disease related, like blood, for example. So just to make you aware that there's also these scores that look at evolutionary conservation. So I mentioned that a lot of the GWAS loci or tag SNPs fall in non-coding regions. A lot of them also fall in tissue-specific enhancers. And in this paper from 2011, there was a quote, "Our results have implications for the interpretation of GWAS. We find that disease variants frequently coincide with enhancer elements specific to a relevant cell type." So on the right here, I'm illustrating that with some data from the Fusion Tissue Project. And this is an image from the UCSC Genome Browser, which is a really beautiful way to integrate a lot of this functional data together. So I have attack-seq peaks up here in certain tissue types. So islets from the pancreas, skeletal muscle, adipose, and then a cell line, GM12878. And you can see in adipose, there's this peak in the attack-seq that is not in the other tissues. And the peak is indicative of open chromatin. And oh, look, the gene is ADIPO-Q, which is the gene that I introduced earlier in the lecture. And it makes adiponectin levels, which is related to adipose. So that makes sense that we might see that there. And if we look, these colorations are the chromatin state from a chrome-HMM model. And the colors are similar to the other figures that I've shown you, with red being more transcription start site promoter regions, orange is enhancer, yellow is a weaker enhancer, green is a transcribed region. So these HASC are human adipocyte stem cells at time 1, time 2, time 3, and 4 in development. So they're kind of pre-adipocyte cells, and then adipose here. And so you can see that these peaks fall in enhancer regions here, and those are upstream of these transcription start site promoter regions, and that makes sense with this gene picture that we see here. And then the GWAS catalog has genome GWAS SNPs that are in this area as well. So this just to illustrate this idea that this is a tissue-specific enhancer. It's in a tissue that is relevant, and the enhancer is only active in this adipose tissue for this gene ADIPO-Q. And yes, zooming in on this region where we have the open chromatin, the enhancer state for the chromatin state for enhancers, the GWAS loci, and the promoter region. Another thing I just want to quickly touch on are massively parallel reporter assays, MPRA. There's a lot of different kinds, and they help us look at something that we might have used to have done on a targeted level, like looking to see if an enhancer area makes a luciferase, if it makes something glow, then it's active. You might do that if you have a hypothesis, you would do it at a targeted level. Massively parallel reporter assays let you do reporter assays like those luciferase assays everywhere, all sorts of places across the genome. And there's one that helps us identify transcriptional enhancers. It's called StarSeq. And essentially, you can look at, if you have an enhancer region that you're interested in and do that genome wide, and you would have reads that come out from the sequencing assay. And here, the most important part that I want you to see is that the luciferase activity on the y-axis and then the StarSeq enrichment on the x-axis are highly correlated for strong enhancers and weak enhancers are here, and then there were some negative controls here that are really highly correlated. It's a nice method. StarSeq is a nice method to do something genome-wide. And there's all sorts of different clever ways to parallelize reporter assays that help us understand activity of these different genome elements like enhancers. So now we can move away from the regulatory regions that we've been focusing on and talk about gene expression. So back to this little cartoon where I have the colored parts of the genome with the exons and introns. I'm also now showing you mRNA up above that has had the introns spliced out. And you can see that in this example, there's fewer RNA molecules, so we would say it's less expressed. These are transcripts, also known as transcripts. And then here there's more transcripts, and so higher gene expression. RNA sequencing is a really nice method using next generation sequencing technology to quantify how many transcripts exist and to identify the sequence of those RNA transcripts in a cell. And there's bulk RNA, which means you're looking at a lot of RNA from a lot of different cells, or there's single cell RNA, which has gained a lot of popularity in the last few years because it really lets you target specific cells, because even in a bulk tissue, there's going to be several different types of cells that might have different expression profiles that you're missing if you do the bulk tissue. And this is a violin plot from the GTEx consortium. And this has been a lovely resource to allow us to look at gene expression across a variety of different tissues that come from cadavers. So back to this ADIPO-Q gene, we see that there is expression on the y-axis is TPM, which is transcripts per million. It's a common metric for normalizing the transcripts by the read sequencing depth so that you can compare across tissue types. So there's expression in adipose, subcutaneous, and visceral, a little bit in the artery, and then in the breast, mammary tissue, a little bit here in nerves. That could be a bit of non-specificity of like the bulk tissue that I was speaking about. Sometimes tissue types will have, like if you take a sample, it could have adipose cells mixed in. So otherwise, this makes a ton of sense that we have, high expression in these tissue types that are adipose, because this is ADIPO-Q, a very adipose-specific protein hormone. And again, as I said, the transcripts per million is a common metric for the y-axis, and you might have also heard it. There's other ones, RPKM, reads per thousand, and reads per 1,000 million. Oh, sorry, reads per kilobase, which is 1,000 per million. So that's another way to normalize for sequencing depth. So gene expression is very interesting because it could be different based on really biologically important things. So the cell or tissue type, we have certain cells that are going to express genes that others aren't. So the gene for making muscle fibers is not necessarily in the brain. You could have differential gene expression because of environment. Our bodies are going to turn genes on and off based on if we've been exercising or eating. So we would have differential expression in genes associated with insulin response, inflammation, or energy metabolism based on what's going on in our environment. We have differential expression by sex. So men's skin cells have a higher expression of genes related to hair growth compared to women's skin cells. And then if we really want to try to make sense of the GWAS loci, we are curious about why we have differential gene expression due to genotype. So what genetic variation, and is it found by GWAS, is affecting gene expression? And that can help us to better understand what the genetic variant is doing. And this is called an EQTL, an expression quantitative trait locus. So there's different methodology that basically looks for a linear regression with the gene expression on the y-axis and then the genotype on the x-axis, and sees to see if there's a pattern between this. And this genotype here, you can see that for AA, in this kind of toy example, has higher gene expression in a number of samples compared to GG. And I've also represented that here in this cartoon. So we would say that was a significant EQTL. We can also look for splice quantitative trait locus, SQTL. And this time, instead of looking at raw expression values, we're looking to see the ratio of isoforms. So this is a gene that has multiple isoforms, which means it's spliced differently. So we have one situation where there's one exon that has been put together, and then there's another situation where 3 exons have been put together. And that's one way that different proteins are ultimately made out of the same genic region or DNA region. So with this genotype GG, we have 100% of isoform 3, whereas we have a mix of the two when we're heterozygous, and then we have 100% isoform 1 when the genotype's AA. So we could put some ratio of the isoforms on the y-axis and again perform a regression to understand the relationship between the genotypes and that splicing ratio. So there's also a whole suite of methods called co-localization methods. So you could like visually look at a region of the genome, and here I'm showing like a genomic position on the x-axis and a GWAS association signal, typical of what we might see in a locus zoom plot, and our lead SNP is here, and then we have an EQTL association in the same area, and look, they look really close. So visually, I'm curious about what's going on there. We can perform co-localization analysis, and we can either see that these are non-co-localizing signals, that because of LD, the linkage disequilibrium, these are two independent SNPs. And so the EQTL does not seem to be actually localized with the GWAS SNP, so it's independent. Or we could say that they co-localize and that this pattern that we saw is actually significant and very likely to be due to the GWAS lead SNP being highly correlated with that EQTL lead SNP, and therefore we would think that this GWAS genetic variation might be influencing our gene expression. And if things co-localize, it could mean causality, which is what I just said, that this single SNP might be influencing the gene expression, or it could be pleiotropy. We could have a single SNP that is doing independent things. In the outcome might be one thing, and the gene expression, EQTL, might be another. Or the GWAS with the outcome would be one thing. So let's think back to kind of putting it all together. We perform a GWAS, we have a Manhattan plot, we have all these loci that we want to understand what's going on, because that ultimately gives us insight into understanding disease pathophysiology or how to find drug targets. We might zoom in and look on the signals with locus zoom plots, try to understand the linkage disequilibrium patterns, understand what genes these variants are near. We might perform annotation. We can look at EQTL and expression, which is what this is illustrating, and we can look at different chromatin states based on tissue-specific methylation patterns in the histones or acetylation patterns on the histones. And then at the end, we might make a hypothesis that, we saw genetic variation in this enhancer. We think some transcription factors binding here. It looks like when there's the G allele, we have more expression of a certain gene. And this is actually a real example of variant to gene to mechanism that had the pattern of decreased H3K27 acetyl increased transcription factor binding, and it was actually a repressor. So it wasn't an enhancer at this point. So that's why there were decreased open chromatin regions. It wasn't as much of an enhancer as a repressor at this point. Decreased enhancer activity, decreased ADCY5, decreased insulin secretion, and increased risk of diabetes. So this is kind of the wild type state, and this is the disease state, which is associated with diabetes. And so this is what we want to do. We want to think about the tissues that the gene is expressed in. We want to think about the causal gene. We want to think about enhancer or repressor activity, what transcription factors are binding, and how does that tie together to the ultimate outcome of our GWAS. So the causal SNP, which SNP is an LD, and is the functional variant. So sometimes our tag SNP, the one that has the most significant p-value from GWAS, may not actually be our causal SNP. Sometimes it might be in high LD, so we want to actually figure out the causal SNP. We want to figure out the target gene, and sometimes there could be more than one target gene, and a causal SNP may be very far from its target gene because of that 3D conformation of the genome that we talked about. We want to think about the relevant tissue. Sometimes the genetic variation is really only relevant in a tissue that is important to that disease, so adipose and obesity, for example. The time is important. A lot of things might be happening during fetal development, which is harder to measure, or an environmental interaction that's going on, like when we eat or when we exercise. And then the direction of effect is important. So when you perform a GWAS, you want to know which of the alleles in the genetic variation is increasing your risk of disease or potentially decreasing your risk of disease or whatever quantitative trait. And then what might be increasing or decreasing a protein or protein function, protein level or function. So we have to understand all of these to understand the mechanism and then therefore the disease and think about treatment, which is, I think for a lot of us is our goal. So this is just a nice way to visualize that we have to think along an axis of the tissue, along the axis of the genetic variation that exists in the genome, and then along the axis of time. So back to this adiponectin, GWAS, and the 7 conditionally distinct signals, we, from our colleagues, Dr. Karen Mulkey at UNC Chapel Hill, they were able to find some evidence for a few of these signals. So the most significant SNP A is actually an EQTL that co-localizes with AdipoQ, and the GWAS signal co-localizes with an EQTL4 AdipoQ, and then this long non-coding RNA, and that's an adipose-specific EQTL. So great, it's in a relevant tissue. E happens to be a splice variant, so it changes how the gene is spliced, what exons and introns are put together. F is another adipose EQTL, and G is actually a missense variant, so that's a little more straightforward. It's a mutation in the coding variant that's affecting the protein, or the amino acid in the protein. So this is the evidence that exists, and it's complicated because we still have a few signals that we're really not sure why are you significant and independent from the other signals, because you're not making sense with all the information we put together. So this is an ongoing process for a lot of diseases and a lot of loci that we really want to understand better. I just want to quickly review a few data sets that I think might be important for you to know about. OnCode, ENCODE is the Encyclopedia of DNA Elements. They have performed a comprehensive parts list of functional elements in the human genome, looking at all the protein and RNA levels, regulatory elements that control cells. And this is a kind of descriptive figure of all the different data sets they have. Not all molecular assays have been performed on all of the tissue types that they have, but they do have, this is the chromatin conformation that we talked about. These are things to look at open chromatin. We didn't touch on FAIR-seq, but it's very similar to DNA-seq and ATAC-seq. They have ChIP-seq data on a variety of different things. They have whole genome bisulfite sequencing. RRBS is another type of sequencing for methylation, and then methyl array, like I mentioned. They've made computational predictions, which you might hear called like in silico functional follow-up, RNA-seq data, and then another type of data that we haven't discussed. But essentially, they've been trying to put together molecular assays for all of these different parts of the genome, regulatory elements, promoters, and transcripts. And if you go to this website, you can look at this giant matrix that shows you for what cell lines and tissues do they have the different molecular assays for. The Roadmap Epigenomics project is human epigenomes for primary cells and tissues. So yeah, so ENCODE is just cell lines. The Roadmap Epigenomics focused on cells and tissues, and really kind of made a reference epigenome. In addition, we have a reference genome. This kind of looked to see what is the basal state of the epigenome? How can we understand gene regulation, cellular differentiation, and human disease? So lots of different tissue types in adults. They even have some fetal tissue types. They have some embryonic stem cell derived tissues, like neuronal progenitors, some iPS cells, induced pluripotent stem cells, some different skin subtypes. So some really high resolution of tissues, which is very biologically important to think about. And so these reference epigenomes that I mentioned, and then the different epigenetic profiles are here in this figure. So 127 different reference epigenomes and all these different data sets, chromatin states, RNA-seq, DNA-seq, different histone modifications, whole genome bisulfite. So again, lots of data that's publicly available for download and available in the UCSC genome browser, which I mentioned earlier, which really helps you, especially if you're a visual person, integrate all the data visually at once and look for some patterns, which could help you build and develop hypotheses. The GCHEX project, which I mentioned, they've measured gene expression with RNA sequencing to see what genes are expressed in what tissues, and they have cadaver donors, and they've done this on a lot of different tissues. They've measured genotypes, genotype arrays, and then went ahead and performed the eQTL analysis for you. So you can download the eQTL data to see genetic variants associated with differential gene expression and in what tissues. And that's all downloadable and I think available on the UCSC genome browser. And just to clarify, because it can be a little confusing, the genetic variant itself is typically called the eQTL, and then the gene that it's associated with can be called the E gene. And so they have this genome-wide, all these pairs of eQTLs and E genes, and an eQTL can be associated with multiple genes, multiple E genes. So you have all these pairs and then across 44 different tissues. And this is just a figure showing the different parts of the body. And this also matches with the violin plot that I showed you earlier, like the different brain regions, adipose types, subcutaneous and visceral, and these colors all match that figure if you want to reference back. And the other nomenclature thing that's important to think about is we typically say cis E genes when we're talking about something that's a local interaction, and then trans E genes when we're talking about something that's distal. Some people have a definition for the trans E genes that also means it's on a different chromosome. So not only is it some X large amount of bases away, that's a distal interaction, but sometimes people also mean it's a completely different chromosome. So a genetic variant on one chromosome is affecting gene expression on another. And that's due, again, to how our DNA and the chromosomes are all packed into the nucleus together. And this is that same color coordination for the different tissue types for the number of samples on the y-axis. So they have fewer brain samples, but quite a lot of adipose and artery and muscle and other relevant tissues depending on what disease you're interested in. The other, more of a software that I wanted to bring your attention to is something called the Polygenic Priority Score, or POPS. And there's basically different ways when you do a GWAS and you get lots of different loci to identify causal genes. So you can think about just what's the closest gene, the nearest gene, or you can think about locus-based methods that think about protein coding variants, distance, enhancers, EQTLs that kind of integrate all that data together. And then there's also similarity-based methods that look for patterns in associated genes and then nominate other genes that have similar functions or pathways or networks. So POPS is a software that allows you to use gene expression pathway databases and protein-protein interaction networks to find high confidence genes that are your causal genes. And this figure is just showing if you used POPS and then comparing to other commonly used methods for nominating causal genes, how likely the top genes were to be represented in the other data. So like for atrial fibrillation, for example, the top 5 genes that POPS found were also the top 5 genes like by distance. And then there was some overlap with some of these other methods. LDL, cholesterol was a little more, the coding variants seemed to also nominate the same genes as POPS. TWAS, different methods. So POPS and then DPICT are 2 methods that allow you to nominate some of the causal genes based on your GWAS, and they integrate a lot of this functional data together. So for some published examples, this is a figure from actually some more fusion data looking at type 2 diabetes GWAS SNPs, and this orange color, these are ataxic peaks in cell line, adipose, and muscle. And so you can see these peaks are quite specific to the muscle samples. And these are peak calls, so basically significant peaks in the ataxic. And then the chrome-HMM was performed to have chromatin states, and there's this chromatin state that is enhancer state that's specific to the skeletal muscle sample. And then it's overlapping this UNC1 region, which it has a lot of different isoforms. And basically there are type 2 diabetes GWAS SNPs that fall in this region that is, that has all these enhancers and the ataxic and the genes. So I'm kind of putting this all together in the UCSC genome browser to make some hypotheses about these type 2 diabetes GWAS SNPs. This is a figure that used the roadmap epigenomics data that I talked about to see if there were patterns in the GWAS loci that were enriched in open chromatin, so ataxic data, in specific tissues. So this inside piece is actually the GWAS p-value, like the colorations. This is the legend for that. And then on this outside is the name of the tissue type. And so the most significant loci are in the fetal heart. And the size of the word, I think it has to do more with the number of samples. So it's a little, your eye goes to these other tissue types, but this was the area that was called significant. So the atrial fibrillation loci were enriched in areas of active chromatin in fetal heart tissue. So this goes back to this idea about timing and potentially the area that this genetic variation is in. It's relevant in a very early developmental time stage, and something regulatory in the active chromatin is going on, and it may predispose you to way later in life having atrial fibrillation. So this is interesting really when we think about that axis of tissue type, time, and the genome. And this is results that come out of DPICT, and there's a lot to unpack here, but essentially we can look at different tissue types where the associations fall that that there's EQTLs for. We can also look at these networks of the gene sets. So there's a lot of significance in the gene set for failure of heart looping and abnormal heart development. And this is also for atrial fibrillation. So this really builds on this hypothesis that there could be some kind of heart development going on. And then here, looking at different histone types, and in the heart, in fetal heart, left and right ventricle and right atrium, there's some enrichment here for different enhancer states going on. So again, DPICT has a lot of different things that it does, but it integrates a lot of the methods, or it's a method that integrates a lot of the data that we've talked about and allows you to move from your GWAS loci to making some hypotheses about what's going on mechanistically and biologically. So we've gone through a lot of information. Essentially, GWAS loci need a lot of work really to understand what's going on. You can't just pop your Manhattan plot in a paper anymore. We want to think about the biology of what's going on. And there's so many high throughput sequencing molecular assays that allow us to do that. So we talked about ATAC-seq, DNA-seq, ChIP-seq, whole genome bisulfite sequencing that allow us to quantify the genetic architecture and look at the sequence in that area. Lots of data sets and tools have been created and made publicly available that help us annotate the GWAS loci. And then I gave you some published examples. So I hope that these slides can serve as a resource for you. And I'm more than happy to answer any questions when we meet in person. And my email is bwolford@umich.edu if you would like to email me beforehand. Thanks.